Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy of my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. A quick note about the foundation. We're embarking on our Anxiety and Depression Codex. It's a resource for people suffering from these conditions or people that know people that are suffering. The premise here is that if you go to an expert, on anxiety and depression, they're likely to have 2 or 3% of all the possible treatments kind of in their, on their wagon that they can help you with. What if we're able to go through thousands of disparate sources and assemble up to 20% of all the possible treatments for this condition? So find out more about this and about donating by going to findinggeniusfoundation.org. And today, my guest is Andriy Marusic. PhD is part of the Marusic Lab at the Moffitt Cancer Center. And we're going to talk about his research. So, Andriy, thanks for coming.
Thank you for having me.
Tell me about your research. What are you working on in the world of cancer?
Yeah, so we are interested in looking at cancer from the perspective of ecology and evolution, in particular, how cancers develop resistance to targeted or cytotoxic therapies. There's been enormous accumulation of knowledge of specific molecular mechanisms within reductionist paradigms. So we understand a lot about the genes and pathways that they control, but this gives kind of good practical, tactical tools, but it doesn't really, this kind of knowledge is not sufficient to predict and understand how tumors will react when we start treating them and how they change. So we're trying to understand the reason and logic underlying these changes. And to understand that requires really applying the concepts from the fields of ecology and evolution to put some sort of foundation for this knowledge. And the idea that once we have this understanding, we might be able to predict more effective ways to interfere. So instead of waging only tactical battles, have more of a strategic plan of how to deal with the disease.
So what does that mean? What are you looking at in terms of tumor environment, tumor metastasis, epithelial to mesenchymal transition? Like, what are you looking at?
Specifically, we're looking how tumors change in response to therapies. For example, what my major focus recently has been understanding how targetable lung cancers develop resistance to therapies. Kind of good poster child example is ALK-positive lung cancers. We have drugs that are extremely effective in shrinking tumors and gaining the short to medium time control over the disease. So we have patients that can stay on the drug for months and years, but almost inevitably advanced cancer relapses at some point as tumors develop resistance. And currently, most of the understanding how this happens is focused on specific molecular mechanisms, such as mutations that occur in the kinase pocket domain of the target of the drug, which basically prevent drug from binding and shutting down the target, or amplification of either primary target or alternative receptor tyrosine kinases that can provide bypass signaling. Or one thing that you've just mentioned, EMT, basically the idea that this transition towards a more plastic state might provide a pathway to resistance. Microenvironment, there is plenty of evidence that microenvironment plays an important role, and it can at least partially desensitize tumor cells to the drug, in some cases even completely remove the sensitivity. And the thing is that we are essentially focusing on one of these things at a time, but in reality, they are happening. All of these mechanisms are operating at the same time in the same tumor cell populations, and sometimes even in the same cells. So in order to make sense of what's going on, we are trying to basically figure out how do we consider all of these factors together. And of course, it's unrealistic to consider everything. The point is to identify the major sort of players, the major factors, and figuring out how to integrate their impact. And to do so, again, it's impossible to do it with just a linear logic. So we're collaborating a lot with mathematical modelers from the Department of Integrative Mathematical Oncology here at Moffitt, where we're trying to create in silico models that describe the behavior of the tumors under treatment, where we basically make the model trying to recapitulate the reality as realistically as possible, and not only to create a sort of in silico version, but also challenge the predictions of the in silico models to see if they can produce testable predictions that could allow us to either refine or validate the models. And realistically, we're not there yet. We're working in this direction, and while we're moving there, we're trying to address some of the lower hanging fruits within this sort of integration paradigm to improve therapies. For example, it's known that microenvironment can protect tumor cells from the drug, and we know that the cells oftentimes develop cell intrinsic resistance mechanisms. There's not much known about how these two interact. Like, what is the fate of a sort of cell that has acquired some partial intrinsic resistance?
What does that mean? Resistance to what? To chemo or to
yes?
What?
So right now,
immune system?
I'm talking primarily about the resistance to targeted therapies, and that's actually a great qualifying question. I should have been sort of more explicit. So, in case of the targetable lung cancer, you have sort of drugs that are taken in a pill form daily, so they have relatively low toxicity, and so patients can take this drug every day, and so it leads to kind of constant presence of the inhibitor in the body, and sort of leads to kind of continuous shutdown of the drug target. And it's quite different from the chemotherapies that are the primary treatment for those cancers where targeted therapies are not available. And the chemotherapies is quite a different game.
What does that mean, targeted therapy? Is it immune therapy, or what is targeted about it?
So the general idea that you have a drug that is directed against a specific molecular target. And so the first sort of, and it can actually mean different things, but the primary application of the term is to indicate therapies directed against oncogenic kinases. And the first targeted therapy that was successfully applied in clinics is inhibitor of BCR-ABL, Gleevec, that enabled management of CLL, CML, chronic myelogenous leukemia. And basically this paradigm opened up a lot of research, and which led to development of similar types of inhibitors in melanoma, BRAF inhibitors, in lung cancer. For EGFR mutant lung cancer, you have EGFR inhibitors such as gefitinib, and now osimertinib is the frontline therapy. So basically the idea that the understanding of the molecular biology of the cancer cell enables to develop drug that specifically shut down abnormal signaling nodes. And if a cancer cell relies on this signaling, the idea that you can kill or stop tumor cells from proliferating and shrink the tumors and gain control over the tumors.
Okay, so how does ecology and evolution come into this? I mean, you're talking about specific mechanisms to inhibit, I guess, the activity of cancer cells, but
yes,
you alluded to the beginning that you're looking at it in a more comprehensive way, right?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
So basically what happens with these great drugs, they get good clinical responses, but they ultimately fail. And there are sort of 2 components for it. One thing is that these therapies do not eliminate all of the tumor cells. So some tumor cells are capable of surviving, and this reflects either their intrinsic properties. The problem is that tumor cells are not exactly identical. There's a phenomenon of intratumor heterogeneity where essentially cells in a tumor are like populations of organisms where you have individual to individual differences, and these differences exist on both genetic level and epigenetic level. And there's also differences in the environment. So some tumor cells are close to, stromal niches where they can get some paracrine signals produced by non-tumor cells within the tumor, and they can sort of get partial protection from the effect of the drug by the virtue of being close to this niche. So that's basically one aspect of the ecology. So it's kind of spatial heterogeneity and the importance of consideration of different environmental niches. There's also kind of interactions between different tumor cell subpopulations. For example, so if a tumor cell starts producing a hepatocyte growth factor, this leads to partial protection by activation of signaling axis, but because this factor is secreted outside of the cell, such a cell can protect not only itself, but its neighbor. So basically, this is consideration of ecology, basically interactions of cells within their environment as well with each other. And then the second part, if we would just like cut the tumor down and retain control, it wouldn't be as bad. The problem is that the surviving tumor cells can change over time. So then they can evolve to therapy-induced pressures. So part of this reflects mutational changes. So every time the tumor cell proliferates, it has a chance to get mutations in the, kinase pocket domain that would dramatically reduce sensitivity of the target to the drug. They can, many other changes that can reduce sensitivity. But then on the other hand, in addition to these mutational stochastic changes, there's a strong evidence that tumor cells can directly respond to the drug and within more of a reprogramming paradigm where the cells with higher degree of plasticity can sort of readjust their phenotype, reprogram their phenotype in a way that puts them in a less drug-sensitive state.
Well, this is a question I've had for a lot of cancer interviewees. Do you see coordination of the cells that comprise the tumor so that the tumor acts as one, or acts as a separate entity, or acts as like a biofilm, as in when bacteria come together?
So I think that it's essential to consider, again, the possibility of interactions and see whether we need to consider these interactions. So again, this is more of for ecological aspect of it. So oftentimes if you have cells that are kind of interacting, forming cell-to-cell context, it becomes more difficult to kill these cells compared to the cells that are dispersed.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So again, I don't think that sort of the biofilm analogy is kind of perfectly applicable, but to an extent. And then again, so it's not one size fits all, even though we have the same...
Do tumors, all the cells of a tumor act together, or do they not? Or do they only act together in certain circumstances in response to stress or niche construction? I mean, which way do you see it?
And that's kind of interesting, the subject that is still kind of a subject of debate among people who look into this thing. So on one hand, there is kind of good argument that tumor cells behave as sort of kind of a Darwinian entity as units of selection, where basically this variability within tumor cells impact their probability of surviving and proliferating. But then on the other hand, again, there's also strong evidence that you cannot really fully understand the tumor unless you consider more of kind of collective behavior and interactions. So in the reality, I think there is a need to consider both sort of both the Darwinian aspect of it and also more of a kind of coordination. And of course, these are not mutually exclusive, but there's not kind of a proper way to really integrate this different angle. And this is kind of one of the challenges that we're trying to address.
You know, if I think of my organs, my liver cells certainly act in concert. They don't act separately. And if a tumor, you know, some people say, well, very few. I mean, I think so. It's like a separate life form that acts, you know, on its own accord. But if it's not, and it's just our cells going rogue, then it's still our cells. And so again, in our different tissues and organs, we see a lot of collective action. Liver cells don't just act on their own. So, I mean, why would it be so strange for a tumor to act on its own? You know, all the cells to act in concert to further the advancement of the tumor. So
touching, I think, extremely important point that is really not very well kind of conceptually developed. So again, like I think it's essential to consider both of these things. So on one hand, I mean, there's kind of good saying that sort of tumors are kind of caricatures of normal tissues. And so even sort of the generation of a tumor, it's not kind of like black and white switch. So you have most tumors, at least the early stages, have kind of represent kind of aberrant forms of normal tissues with kind of similar structural units. And then as the tumors progress toward more dangerous, invasive metastatic disease, usually there is at least some of these tissue characteristics are partially lost, but then they're not lost entirely. And so to understand the tumor behavior, you have to, one needs to still to account this developmental program that can be executed within these tumor cells that basically reflect their normal tissue lineage heritage. On the other hand, you also have the Darwinian dynamics operating there, where again, like the tumor cells with certain phenotypic changes can have advantage over others with and sort of take out the space and outcompete them and get expanded. And basically, this is essentially what's happening, part of what's happening under therapy. So eventually, we're selecting tumor cells that are no longer sensitive to the drug, and they can sort of thrive and proliferate even in the presence of the treatment. But to my understanding, it's not possible to completely account for what's going on by applying only this competitive dynamics and looking at tumor cells as sort of individuals that act on their own.
Are you trying to model it as if they act on their own, or are you trying to model it from a Darwinian perspective, or are you just observing these things? Like, what's your goal?
So our goal, like, again, we're starting with experimental system where we're trying to have an open mind and figure out what is going on. And so we start with sort of the simplest assumption and simplest models. And then if the reality of sort of trying to sort of set up the experiment to address, can we explain the behavior of the system with these simple assumptions? And basically, if the results tell otherwise, then we have to revise those assumptions and consider interactions between the subpopulations. For example, in one specific example, again, it's a simple experimental system where we cultured two types of tumor cells together and are setting up the essentially, this is kind of the parental work of my collaborator, great scientist, physician, mathematician, Jake Scott from Cleveland Clinic, and Artem Kaznachev, his students, were basically trying to kind of infer the existence or lack thereof of interactions between these two types of tumor cells from the observable experimental setup, and then basically try to sort of understand the nature of interaction, not on the molecular level, but on the level of the impact of coexistence of these cells, whether together they behave differently compared to sort of some of the parts when these cells are separate.
What have you been able to figure out about tumor behavior? Anything yet, or is it still early in your investigation? Like what interesting insights have you gotten?
In sort of the experimental system where we spent most of our attention, there are a couple of very interesting things that we have found out. So So we started, again, looking at how tumor cells develop resistance to ALK inhibitors. And this is one of the targeted therapies that is administered continuously, very effective, but eventually failing in metastatic advanced cancers. So usually people are thinking of resistance as reflecting a single change, either it's a mutational change or change in expression of certain gene or EMT, etc. And so we found actually that we cannot. We have this experimental system where we can get quite predictably the tumors, population of tumor cells in vitro or in vivo eventually develop resistance within meaningful time frame. And what we found is that we cannot really explain this resistance with any single mechanism. Instead, resistance appears to reflect a combined output of multiple individual changes. Some of these changes are induced by the drug, and other changes are stochastic, and they can act in additive and synergistic ways. And so this is kind of, I think, a very important observation that some people have made before, but without, I think, paying due attention to it, because I think it has a profound implication. So, for example, one of the problems with these relapsing tumors is that despite all of the research, all the papers that look at the resistance mechanism, typically once the tumor starts relapsing, even if you target this mechanism that is associated with resistance, in most cases you get like very transient response. And a part of it is that the tumors are heterogeneous. You might sort of suppress a subpopulation of cells with this targeted mechanism, but then the other cells can grow out. But where our result suggests that it's okay if you have multiple mechanisms that give rise to resistance and you take one out, even if it's a dominant one, you still have cells that are more robust than the treatment naive cells. And so they'll have easier time figuring out alternatives compared to if they have to start from the square one. So this is one recent discovery that I have that I think would have a profound implication of how tumors develop resistance. Another thing.
What does that mean? Like, I don't understand. What are you looking at with this? Are you specifically targeting cells that have been changed in a particular way and biopsying them and seeing their pathway changes? Or I don't know how this plays in.
And that paper, that study that I'm referring to, published it recently in Nature Communications journal. So in that case, we're looking at cells in vitro. So we start with therapy naive cells, the cells that haven't seen the drug. And then we're looking at how they respond to different clinically relevant drugs that inhibit this key oncogenic change that defines these cells. And so what we're finding that consistently, so the cells can develop resistance to any of the 4 different inhibitors that we have been testing. And because this is an in vitro study, we can actually sample and look at the different time points, looking how tumor cells change. Not only looking at the before and after, but also looking at the intermediate stages. And so initially, again, we did not start the project expecting to see this complex multifactorial resistance. We started by identifying what are the known resistance mechanisms that we can see in resistant cells. And we found, instead of one, we found multiple ones, and like knocking out any one of those couldn't really fully restore the sensitivity to the original. But when we started kind of combining things, we see now the evidence that basically resistance reflects contribution of multiple individual molecular changes rather than a single factor.
Well, is there a predominance to the changes? Like, how would you characterize the changes that occur in a tumor? Is this before and after chemo, or again, under what conditions? It just sounds a bit general. I don't understand.
Yeah. So in this, this is where we're dealing with targeted therapy that is applied continuously. So in context of in vitro studies, we're basically putting a drug into the media, and then basically we replenish the media twice per week, and we're adding sort of new drug with new media. So basically the tumor cells are exposed to kind of essentially continuous presence of the drug. And so what we're seeing immediately, as soon as you add the drug, within a couple of hours, you have some of the tumor cells start dying, other tumor cells arrest their proliferation, but then like over time, they can better and better and better until they get to the point where they can ignore the drug. So, but as early as like within 4 hours that we hit them with the drug, we're already see changes in genes like HER2 and EGFR, and these are alternative receptor tyrosine kinases, and their expression can make the tumor cells less sensitive. And so because there's this change happens so fast, it cannot really reflect Darwinian dynamic sort of variability and selection. So this is an example of direct drug-induced response of the tumor cells. And it looks like not all of the tumor cells are capable of making these changes, but those tumor cells that can make this change, they're capable of at least hanging there for some time. And then if we look sort of further down the line, we see now kind of expansion of certain subpopulations that carry out certain stochastic changes, such as amplification of the original target, AML4ALK, where now in crizotinib, one of the drugs, we're seeing that inevitably we see an expansion of tumor cells that have this target amplification. And in the literature, it's considered that this target amplification can confer resistance to the drug. And so if a clinician sees that the tumor became sort of resistant tumor, has multiple copies of AML4ALK, the conclusion is that, okay, so this amplification has conferred resistance, but basically what we're finding is that by itself, it cannot fully explain the resistance. We can artificially recapitulate this scenario of AML4 amplification by integrating additional copies of the oncogene into the cells using lentiviruses. And we are seeing that despite phenocopying the signaling effect of the endogenously observed amplification, these cells are only weakly resistant, and it sort of takes these additional changes, some of which are stochastic and some of which are induced, to get to the full degree of resistance. So basically getting to resistance is a process. It's a trajectory rather than a simple on and off switch that can be conferred by a single mutation.
How do you know a change is stochastic or directed?
So,
how could you tell?
And this sort of example, so if we see change as early as like 4 hours out of the treatment, and at this point it's a time scale that is too small for cells to proliferate, where you have a rare subpopulation that expands, it's sort of clear that this is an example of induced response. However, for mutational changes, if you're talking about the point mutation in the kinase pocket domain or gene amplification, I think there's like decades and decades of research that clearly show that these kind of changes are stochastic. They're more rare. They're not really directional, but they can have a profound impact on the sensitivity of tumor cells. So every time that a cell divides in the process of DNA replication, you have dozens of small-scale point mutations, and most of these point mutations don't really matter. So the probability of mutation of specific nucleotide is very low. But then, if you have large population of cells and many rounds of proliferation, eventually you will get some of these mutations to pop up. And it was essentially the basis of the mathematical argument that resistance must pre-exist in the tumor cells before the treatment. But basically, what we're seeing that even if we do have these mutations pre-existing, they're not fully resistant, and the cells have to acquire additional changes to gain the full extent of resistance. Also, their ability to get there cannot be fully understood if you only focus on these cells. You have to consider the environment and the effect of competition with other cells. For example, if you have the treatment that kind of completely eliminate all the tumor cells that only have this intrinsically resistant cells, these cells can expand and take over the tumor very quickly. However, back to this phenomenon of microenvironmental resistance, we're seeing that if there are enough tumor cells that survive, not because they're intrinsically resistant to these tumors, but because they are in the proper niches, the intrinsically resistant cell still has to kind of outcompete those other tumor cells to expand and take over. And this is basically goes to this aspect of importance of integration of multiple levels of consideration, where you have to think about both this induced epigenetic changes, about the stochastic mutational changes, about the microenvironment, about the impact of competition between different tumor cells within the tumor.
Is there a way to design an experiment so that you can see what is a deliberate adaptation versus a stochastic one? It doesn't seem like there is any stochastic stuff. I mean, it seems like adaptation that can lead to maladaptation, therefore it's not successful, or it can be successful.
Well, I think there's kind of very, very strong evidence that the mutations are stochastic, whether you're talking about the chromosomal amplification of piece of segment of DNA or specific point mutations. So these are stochastic changes.
But how can they be if they reoccur in a given cancer? Not all of them, but I'm sure a vast majority of them reoccur and have been characterized for a given cancer.
But this is kind of more of a kind of basic Darwinian dynamic. If you're dealing with low probability events, but that you have a population consisting of millions of cells, and then every time the cell replicates their DNA, there is a chance that this mutation occurs. And so if the mutation occurs and it gives the cell a fitness advantage, it will be amplified by selection. So this cell would have they will expand at the sort of expense of those tumor cells that don't have this adaptation. And so basically that is the reason where we're seeing this recurrent mutation is not that it's a reflection of a directed response, but because like if you have even low probability event, but in large enough population over time, some cells would acquire these mutational events. And if these mutational events confer substantial advantage, the cells.
That's what everyone says about Darwinian evolution. But again, cancers appear to have tropisms for certain tissues. Even though they're heterogeneous, they still seem to have a common set of what we call mutations. So if that's happened thousands and tens of thousands of times over various cancers, it seems like a directed thing, even though it may be beneficial or not beneficial for the tumor. But I mean, if it recurs, why would it be random?
Again, like sort of if we're talking about sort of tropism to different tissues, you look at sort of the kind of more of a global phenotype. So they kind of in every sort of cell in a normal tissue, in order sort of to survive and proliferate, it needs to receive certain signals from the environments, basically interactions with the neighboring cell, interactions with extracellular matrix, certain growth factors. If you don't have the signal, the cells either arrest their proliferation and die, depending on what cell type you're. If you're talking about hematopoietic cell, the default response is suicide. If you're talking about structural cells like fibroblasts, the default response is getting into arrest. And tumor cells, even though they are abnormal, they retain at least partially dependence on this growth factor. And that's why, like, in order to grow tumor cells in culture, we have to provide them with FBS, fetal bovine serum, to provide these growth factors that enable tumor cells to proliferate and grow. And then if a cell gets into a different organ, now it's like facing very different context, and it doesn't necessarily recognize the new environmental system. And so in some tissues, the context might be sort of the closer to or more similar to adapt to compared to other systems. And in this case, again, like adaptation, you cannot fully understand it from the sort of stochastical Darwinian dynamics, but it is part of the answer. And another part of the answer is just the ability of cells to recognize certain thing and sort of rewire itself towards a phenotypic state that would enable it to kind of exist in this new environment. It's kind of the term that's referred as phenotypic plasticity. But back to your sort of argument that because we're seeing recurrent mutations in cancer, it cannot be stochastic. One thing is that even if you look at the same type of cancer, they are basically, these things are probabilistic. So it's not that in every cancer, like lung cancer, you have EGFR mutation and ALK mutation or RAS mutation. There are tumors with one set of mutation, and then if you take another tumor, you wouldn't be identical. So it would be kind of a different set of mutation. The end effect would be similar.
But that's what I'm saying is maybe that's how you know something is stochastic or not, is the core set. I'm guessing it's probably 80% of the mutations of a given cancer show up multiple times or many, many times. So I would think a vast majority of them are not stochastic because they show up over and over and over. But maybe there's a small subset, a minority that are stochastic. But again, it's not like I've in all of time, for instance, there's been, let's say, 120 billion people, and they've all pretty much had a liver. That's you can never say that's a stochastic thing.
No, but liver,
so again,
you start with the liver. So I mean,
why is it different though? Why isn't there stochasticity in creating livers or hearts or that produces something different? I mean, maybe there's a little bit of minority, but in tumors, again, like what do you think the percentage of deliberate versus, random changes?
Well, I'm not sure like if it's going to be meaningful, if I can give meaningful answer about sort of percentages. I can sort of say with confidence that basically there are some of the changes that are more of a kind of deliberate or sort of kind of programmed, the kind of where it's sort of part of the cellular identity to respond to a certain stimulus in a certain direction. But other things, in your sort of liver example, it's not that you have people without liver and then like it can appear out of nowhere stochastically. But in case of specific mutational events, okay, you start with like normal one or diploid genome, so 2 copies or say like genomic loci that contains RAS gene. And then if you have sort of cells where so basically in order to get more copies, it requires an additional event, something sort of to happen that is different from the default. And so it could happen 2 ways. One thing, just it happens stochastically, randomly with certain probability. And then if that event gives an advantage, the cell could expand and you would start seeing this more often if the event gives advantage. If it's directionally, you would expect that all tumors would show up with this amplification. And actually in our studies, there was like a very interesting observation where in context of cells that develop resistance to lorlatinib, 3rd generation ALK inhibitor, we're seeing that their kind of global phenotypes are fairly similar and different from cells that develop resistance to alectinib or say crizotinib. But this and in most of these cells, we're seeing evidence of RAS amplification, but in some cells we see amplification of KRAS, and in other cells we see amplification of NRAS. So this is kind of an example of convergent evolution. So you have different stochastic events that basically lead to similar phenotypes. So one sort of biological replicate, where I assume that we have dealt sort of with rare event that produced the additional copy of NRAS, and this having an additional copy of NRAS gave the cells that acquired this event of an advantage and enabled it to expand and take over the population. And in other case, the stochastic event was in sort of parallel, but a different gene that gave a similar phenotypic impact. And so this kind of observation of the convergent evolution, I think provides a fairly strong indication that we're dealing with stochastic events that can be triaged by and discriminated by selection. And then experimentally, what we can do and what we are doing is that we can introduce additional copy of, say, like RAS or EML4 ALK, and basically see if we induce just this change, how does the cell fare compared to the cells without this change? Does it give advantage? Does it give disadvantage? And basically what we're seeing is that the extent of the advantage that is given by these changes oftentimes depends sort of on the history of the cell. So if you give certain things at the beginning, for example, if you give EML4 ALK amplification at the beginning of treatment with lorlatinib, it appears to, if anything, gives a disadvantage to the cells, but then at some point the same event can provide an advantage. And so basically it just tells that the same kind of molecular event is evaluated by selection, and the impact is dependent on the sort of what is the starting point of a cell where the change has occurred. I hope it makes sense, but I'm not sure if...
We're out of time, but what's the best way for people to find out more about your work? Where can they go?
It's a good question. I don't know. We have a brief description on our lab's website, periodically trying to write not just research papers, but opinions, perspective, or describe our inferences and sort of the implications of what we find experimentally. And I think that would be probably the best place to find. And of course, this is not necessarily, it's more of a kind of for scientific colleagues audiences. And actually, this podcast I thought would be kind of an opportunity to really explain a little bit more of what we're doing. But I'm not sure, like I sort of succeeded there because we quickly get into a little bit of a deep...
I think we got into some important details. I mean, I don't think I think it worked well.
Yeah, thank you. But I mean, it's kind of, we're dealing with sort of the ideas that are not necessarily kind of on the surface. And so I understand sort of even like explaining things, it takes, to scientific colleagues, it oftentimes takes sort of multiple time sitting together and going over different pieces until sort of a more kind of clear picture emerges.
Yeah, it's okay. No problem, Andrew. I think it was good, and thank you for coming on the podcast.
Well, thank you for having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.